on metabolic processes. So metabolism is a complex of chemical reactions. Chemical reactions are catalyzed either by enzymes or ribozymes. All of the chemical reactions associated with physiological unknown 1 are catalyzed by enzymes. Don't have any ribozyme activity to speak of. When we're dealing with bacteria growing in vitro, oftentimes we can get a hint of what's going on with their metabolism by looking at either what kind of end products they produce. So they'll form something and it'll be trapped in the medium, or something will disappear from the medium that used to be there. Either way, we can often find out what's going on by putting pH indicators into the media, and so we have a whole bunch of pH indicators associated with this section. It's kind of fun because it makes the enzymatic test colorful. Well, it's fun if you're not colorblind. If you're colorblind, it's a real pain. So, these enzymatic tests that we're going over, they're designed to demonstrate specific aspects of microbial metabolism, and actually they're primarily applied to gram-negative fermentative organisms. However, some of these tests can also be applied to other organism types, so they will have some application to your semester project when you get to that point. So some of you may have organisms you can think about, what kind of tests am I going to need to do for the semester project organisms? Well, as we go through these, I'll point out some that you can. Now, the other thing about this, since metabolism involves enzymes and enzymes are proteins, this also tells you something about microbial genetics. Because in order to produce a protein, they have to have the gene that encodes that particular protein, or sometimes more than one. So we're learning a little bit about phenotype. Phenotype is physiological characteristics. We're also learning a little bit about genotype. Genotype is what are the genes present within organisms. How many people are familiar with the terms phenotype and genotype? So very often people think about these as they apply to humans. For humans, genotype is often something that you can see associated with an individual organism, such as eye color and hair color. With bacteria, phenotype is much more often something that you see because of the metabolic process. Because really, when you look at bacteria, you don't get to see much. I mean, you get to see, are they gram-positive or gram-negative? Can they make a capsule or not? That's part of their phenotype. Do they make endospores or not? But then we start looking at metabolic processes. This tells us more about their physiology. What can they do? And again, all the enzymes are encoded by genes. Okay, so this is kind of a review. This reminds me on the lab quiz for Thursday. Lab quiz Thursday is going to be use of selective and differential media and replica plating and microbial numbers and the enzymatic test that we're looking at the results for tonight. So this is kind of a review. This is part of the use of selective and differential media. So we're looking at 3 different types of media here that involve catabolism of some kind of carbohydrate. So carbohydrate catabolism is often associated with a change in color because of pH indicators. The McConkey's agar, what's the carbohydrate in there? You need to know that. Lactose. The same is true for Churchtol 7, both McConkey's and Churchtol 7. These are media that are differential on the basis of lactose fermentation. If the organisms can ferment lactose, they're going to produce acid, and the acid is going to cause a pH indicator to change color. In McConkey's, the pH indicator is neutral red. In T7, what's the pH indicator? Anybody? It's bromethymol blue. Green when it's neutral, it's yellow when it's acidic, blue when it's alkaline. The other plate, the plate on the right hand is mannitol salt agar. What's the carbohydrate in mannitol salt agar? Mannitol. It's in the name. Mannitol. The salt isn't a carbohydrate. Anyway, the pH indicator in mannitol salt agar. Anybody? Phenol red. If the organisms produce acid, they turn it yellow. If they don't produce acid, actually, if they produce alkaline end products, they'll turn it a hot pink color. It gets a lot pinker than what you see on that plate. So neutral red, bromethymol blue, phenol red. Those are the pH indicators. We're going to use primarily bromethymol blue and phenol red for our enzymatic testing. One other pH indicator, again, you're going to see tonight is brome producible purple, but you already saw that one too with our BCP lactose quiz. Okay, so chemoheterotrophs, they can catabolize carbohydrates using either a fermentation or a respiratory type of metabolism. Anybody remember the test that we used to determine, do they ferment or are they respiratory? Some of you got tubes, I gave them to you last lab. The OF test, the oxidation fermentation test. Okay, so if we want to know, does an organism ferment or is it respiratory, we can run an oxidation fermentation test or OF test. This is very useful for gram-negative cultures. It doesn't work that well for gram-positives, and I think there's something about the bromthiamyl blue, which is the pH indicator. Typically, if we want to know if gram-positives can ferment, we run an MRBT. So this is primarily for gram-negatives, but it is very applicable to semester project organisms. If you have a gram-negative for your semester project, run an OF, because you want to know, can they ferment or are they respiratory? So all gram-negatives, you get to do this. Now, some of you already had the data last time. Some of you are looking at the data tonight. This is referring you to a specific location in the laboratory, these worksheets. I don't ask you guys to turn these in, but it is a good idea to fill them out because they are part of the educational process. So if you look at that worksheet on page 179, the oxidation fermentation test, it asks you, what is the carbohydrate present? The carbohydrate is glucose. The reason we use glucose is because if an organism can ferment anything, it can probably ferment glucose. This is one of the most abundant carbohydrates in existence. So if they can ferment anything, they can probably ferment glucose. The pH indicators, bromthiamyl blue, there you see the color change. It's neutral. When it's neutral, it's green. When it's acidic, it's yellow. When it's alkaline, it's blue. And what we want to see is if the organisms can ferment, fermentation is anaerobic, right? So that means they can grow in an anaerobic environment. If they ferment and produce acid, they should turn the medium yellow. Now, a couple of things to remember. The organisms that are used for the physiological unknown 1 set are heterofermentative. What does that mean? So they can ferment glucose and they can produce more than one byproduct. Hetero means different. They can produce a wide variety of end products. If they produce acid, don't assume it's lactic acid because they can produce a whole bunch of different acids and you don't know which one it is. If they produce gas, it could be CO2, it could be hydrogen. It's not oxygen, but anyway, so heterofermentative, they're going to produce a wide variety of end products. What we're going to be testing for in the OF test is acid production. But sometimes bacteria produce neutral end products, such as acetone or acetylmethylcarbinol, or they might produce alcohol. You're not going to be able to tell that, but anyway. Okay, so this is what the medium started out, looked like, 2 green tubes. You had 2 tubes, one has a Durham tube in it, one did not. And after you guys inoculated them, you left them in the basket. I came in and I put a Vaspar seal on. Actually, there's a little bit of a difference because both of my tubes there have white caps, and the OF tubes that have been given to you guys, one has a white cap and one has a green cap. They put the different colored caps on to tell you which one has the Durham tube in it. I don't remember which one has the Durham tube.
Green one.
Okay, so the green cap has the Durham tube, the white cap does not. In this image, here you can see the Durham tube in this tube. This has been inoculated and the Vaspar seal has been applied. So that's what they would have looked like after you inoculated them. I put the Vaspar on before they went into the incubator. Which reminds me, culture number 16 and culture number 25, I pulled those tubes out. They were not supposed to go into the incubator. Those guys are supposed to be grown at room temp. So they spent the weekend in the box. Anyway, don't put them in the incubator. Okay, so here we see the oxidation fermentation test after a period of fermentation. So these organisms have been growing. This is supposed to be a 48-hour test. Yours have been growing longer than 48 hours, but it won't hurt anything. What we see happening here is the production of what to change the color of the indicator from green to yellow. What are they making? They're making acid. So acid is going to change the color of the pH indicator. Because they're making acid under that VASPAR seal, this is going to keep oxygen out. That means these guys have what kind of gas requirement? Are they obligately anaerobic? No, we already said we can't grow anything that's obligately anaerobic in here because they started out growing on a plate that was in your lab drawer or in the incubator. It's exposed to the oxygen in the air. If they're not obligately anaerobic, they are facultative. These guys are facultatively anaerobic. And their metabolism is fermentative because they are growing underneath this VASPAR seal. Fermentation doesn't require oxygen. Cellular respiration does. Okay, so they're fermenting, they're producing acid. Notice there's a little bit of green inside the Durham tube. Typically that just means that the acid they've produced hasn't diffused into the Durham tube yet. The tubes that I gave people on Thursday last week, several of them had green inside the Durham tubes. I doubt if any of yours do. So if you look at your OF tubes, what color are they? Yellow. They're yellow. They should both be yellow. So that tells you your culture is fermentative. It's producing acid. Specifically, it's fermenting glucose. Are they producing anything other than acid? And how do we know? What else do you look for? David's going to tell us here.
We know it's producing gas.
Ah, we know it's producing gas. Okay, so if they're growing under the VASPAR, they're fermentative. They're facultative. Oops. Well, we just jumped to the side. So yes, they produce gas. Let me back the truck up here. Go back. Go back. There. See this gap? That's gas production. This is significant because you want to know, can your organism produce gas from glucose fermentation? So make sure you record this if you have gas production. If the VASPAR seal is lifted, that's gas production. If there's bubbles inside the Durham tube, that's gas production. That's why that thing is in there, to try to catch gas. But you're going to catch a lot more of it under the VASPAR because it can't get out. Who has some nice gas we can look at? Okay, so here's one. There's some nice bubbles underneath. So we know they're producing gas. Anybody have one? Oh yeah, look at this one. Look at the position of the VASPAR. That's gas production. Okay, the VASPAR goes in as liquid. I can't pour it in there and have it hang in the air like that. So that's gas production. These guys are really gassy. Your organisms are gassy. So if you have two yellow tubes, you know they're producing acid. And if the VASPAR is lifted or there's bubbles underneath it, you know they're producing gas. And specifically, it's gas from glucose fermentation. Again, these guys are facultatively anaerobic. That's a gas requirement. That's not metabolism. Metabolism is either fermentative or respiratory. If they're fermentative, both tubes are yellow, and they grow under the VASPAR. If you get this result, where this is green under the VASPAR, these guys are respiratory. They can't ferment, so they can't grow under that VASPAR seal. There's no oxygen. None of you that inoculated on Thursday should see that. But several people that had 2 gram negatives saw that last week. Some of the cultures that are respiratory produce aerobic acid. This aerobic acid is coming from glycolysis in the Krebs cycle. So think about glycolysis. You have a number of acids that are produced along the way on that pathway. And on the Krebs cycle, you have a whole bunch of acids that are produced. Some of those can get dumped into the environment. So this is aerobic acid production. If these tubes are left to sit for a long period of time, that yellow will tend to diffuse down into the medium. But we had several people that had respiratory organisms that had a yellow top on the open tube. And then this one, we had one example that showed this. Instead of a yellow top, we have a blue top on the open tube. These organisms are using the peptone in the medium, and they're producing alkaline end product, probably amines. So this blue color says no acid production, but they're producing some kind of alkaline end product. Okay, again, the respiratory cultures we saw last week. So if they can't grow under the Vaspar seal, they're respiratory. They're going to be using an oxidative type of metabolism. What might we assume about their gas requirements? If they grew under the Vaspar, we said they were facultatively anaerobic. If they can't grow under the Vaspar, their gas requirements are obligate aerobic. They're obligately aerobic. We could assume that about them, and it might be true. So if the organisms can only grow when oxygen is available to them, we say they're obligately aerobic. It is possible that the organisms could be respiratory and not obligate aerobes if they can use something other than oxygen as a final electron acceptor. We can't determine that with the OF test. And then this is just showing you the gas, and again reminding you the gas can either be hydrogen or carbon dioxide. We don't know which. Now, these heterofermentative organisms, as I said, they can produce a wide variety of end products. Apparently in industrial microbiology, organisms are referred to as being fermenters of a particular substance. And it's kind of confusing because they refer to them as being mixed acid fermenters if they produce mixed acids. And they're called butanediol fermenters if they produce butanediol. To me, this is confusing because what they're fermenting is glucose. They're glucose fermenters. They're mixed acid producers, or they're butanediol producers. That's not what they're fermenting. But the thing is, in industrial microbiology, they're more interested in these massive amounts of end product that the organisms are producing. So their terminology is a little bit confusing. Anyway, don't forget, most of the time, if you're looking at the OF test, you're looking at glucose being fermented, and then the test that has specifically to do with this mixed acid fermentation or butanediol fermentation, this is the methyl red Vogts-Proskauer test. And again, what they're fermenting is glucose. Glucose is the carbohydrate. So this is a test that many of you have, just one of. Actually, everybody should have just one, except some of you didn't have cultures separated enough to do this test. So what you were supposed to do was put a visible blob of cells into a tube of broth, the MRVP broth. Remember, if you're looking at these things, do not tip those tubes sideways and do not hold them by the caps, because they can slip right out of that cap. Hold them by the glass. Always hold them by the glass. So what do you think the difference is between these two tubes?
Turbidity.
Turbidity, yes. And what does this indicate? They grew. When you inoculated it, the liquid was clear. You put an amount of cellular material in there. If you have a lot of turbidity at this point, that means they grew. The culture grew here, not so much there. If your MRVP medium, after the length of time that we've had them in there, which is more than 48 hours, if your medium is still clear, something's wrong. They should not be. What? Well, they may be sitting on the bottom.
They are.
Especially the staph cultures. Some of you had staph.
Oh, there it is.
Please don't tip that tube, Justin. Keep it vertical.
Oh, yeah, there it is.
You're scaring me back there. Yeah, be careful with these. Your culture may be on the bottom, but don't shake them. They leak. Okay, turbidity indicates growth. Now, this is called the methyl red Vogts-Proskauer test. It is methyl red Vogt Proskauer medium, but it turns out these two tests are separate. They're testing for different things. So the first one we're going to talk about is the Vogt Proskauer test. The Vogt Proskauer test is testing for the presence of a neutral fermentation product called acetylmethylcarbinol. You can't just look at it and see it. Instead, what you have to do is take 1,000 microliters of your culture out of the tube that it's currently in and put it in a screw top tube. How did you guys measure 1,000 microliters the last time you did this? With a pipette and a bulb. It was a lot of fun, right? No, nobody liked that. The good news is, for today's exercise, I'm not going to ask you to do that. Yay! Instead, you're going to use digital pipettes.
No, he's not.
I would like to keep these digital pipettes up front so that they don't just get randomly distributed around the lab.
I'm good at labs. I'm just lazy.
The digital pipettes, if you recall, to use these things, you have to put a tip on. To put the tip on, put the pipette in the tip and tap it. Like you would tap a tiny little hammer. Don't stick it in the box and lean on it, because you might break it. When this tip is on properly, there's a gap here. Don't try to close it. Now, to use this, first of all, you want to make sure that it's measuring the right volume, which this isn't, because this says it's going to measure 500 microliters. Actually, it says it's going to measure 300 microliters, and I want to measure 1,000. To make it change, you press this little button. If you're familiar with a standard transmission on an automobile, you don't change gears without putting the clutch in. And that's what this is like. Because if you try to change gears without putting that in, the pipette will make a terrible noise. And your instructor will screech at you, "Stop it!" Because it's very damaging. So I'm just turning this now with the button compressed. I have to change this to read 1,000. Which is a pretty good trick, actually, because I don't have my glasses on. I can't see this thing. Anyway, so now it's going to measure 1,000. What I do with this is put the tip into the liquid I want to measure. I press this plunger to the first stop. When you guys were using the pipettes with the bulbs, remember I said you have to use finesse? You still have to use finesse. Feel that thing. Don't just push it down. Because if you push it down, you don't know what volume you're going to be using. You push it to the first stop. You want to feel that. You can hear it. There's also a little line out here that shows you where it should stop. So you push it to the first stop, you pick it up slowly, and it will automatically pick up 1,000 microliters into this tip. Then you're going to transfer that to a screw top tube, which I don't have any up here right now, but you put it in, you press this to the second stop, and it will push an air bubble out and push that liquid out of the tip. If you push it to the third stop, the tip comes off. Okay, so you're going to measure 1,000 microliters, make sure the pipette is set at 1,000, and then use it properly. You're going to transfer your liquid into a screw top tube. It says these screw top tubes are supposed to be clean. The screw top tubes we have look kind of scruffy on the inside, and it's because we keep putting potassium hydroxide in them. Never mind, it doesn't matter. After you put your culture into the screw top tube, you're going to add 18 drops of Barrett's reagent A and 18 drops of Barrett's reagent B. These reagents are here, so I'll put them up here with the pipettes. And then usually what we do is set up a little piece of particle board with aluminum foil on it, so that these reagents can stay on the foil. This is because we're using a couple of things here that are somewhat nasty. Well, okay, so this is Barrett's reagent A. It's alpha naphthol in ethanol. Alpha naphthol can be a respiratory irritant, so be a little bit careful with that one. And this one is 40% potassium hydroxide. 40% potassium hydroxide will blister your skin if you leave it on there. So if you get this on your skin, make sure you wash it with lots of water. It'll feel slimy. You keep washing it until you don't feel that slime anymore. This is dangerous. It's rather hazardous stuff. So you're gonna put 18 drops of Barrett's A and 18 drops of Barrett's B into the tube where you put your culture. It really doesn't matter what order you put them in as long as you put them both in there. And then you're supposed to close that screw top tube so that it won't leak. The screw top tubes had cork inside the cap. It used to be waxed so that when you screwed it down tight, it was watertight. They've been through the autoclave so many times now, there's no wax left. That's why this says wrap it with a paper towel. So after you've got your Barrett's reagent A and B in the tube, I recommend that you take a little piece of paper towel and just wrap it around that cap so that, because you're gonna shake it, you don't wanna let that potassium hydroxide get on your skin. Actually, what we found is we used to have to tell students to shake it by holding it like this and shake it for a minute. And they get tired of shaking it. And then we decided, actually the easiest thing to do is put it on a vortex mixer. So we'll put the vortexers out.
I have been on this for the lab portion, so we need the space.
That's all right, I'm gonna put this back here. No, I keep getting in your way.
I don't know.
That's okay, I'm gonna put one of these on the other side too. So this is a vortex mixer. This thing is supposed to be set at the maximum vibration, and if you put it on, it just vibrates. So instead, we put it on automatic, then it will only vibrate. Touch it. Where does it go when you touch it? Doesn't do that. Well, okay, you have to turn it on. Anyway, it's gonna vibrate pretty good. The newfangled one. I like the one better. So we'll have that one over here where you have to turn it on to use it. And we'll put this one on the other side.
We only do 1 minute.
She should move faster.
Technically, like we're still at night.
So this one, again, maximum vibration. This one, put it on touch. Now if you touch it, it vibrates. If you're not touching it, it doesn't. So you just hold the tube against that thing and vibrate it good for 1 minute. Then you're supposed to sit it down, just put it back in the rack and let it rest for about, well, 10 to 15 minutes, doesn't matter too much. What's important is that you put it back on the vortexer and vibrate it again. And it's during the second shaking that you're gonna get the color change. If the medium turns red, sometimes this is described as wine red, but if it, usually it looks more like raspberry Kool-Aid. It's really a pinky red. You get that pink red coloration, that's a positive VP test. That means your culture is fermenting glucose and it's producing acetoin. Acetoin is not acetone, spelled funny and pronounced funny. It's not acetone. It's acetoin. It's a neutral fermentation product. It's also called acetylmethylcarbinol. So this and that are the same thing, but this is not acetone. Don't call it acetone. Okay. If it turns kind of a greenish yellow brown kind of ugly color, it's negative VP. So pink red is positive VP, yellow brown negative VP. Okay, after you've done that, after you've set up your your Voges-Proskauer test, the next thing you're going to read is the results for the methyl red test. The methyl red test, you're going to be looking at the liquid that's left in the tube that you started with. Go to there. Here's our methyl red test. So all you're going to do is put methyl red indicator. Why is that not changing? That's weird. Huh. It's changing here. I'm looking at it and going, what's going on? I don't know. Oh man, yeah, don't you love technology? Like, come on, computer, pay attention to what's going on.
Yeah.
All right. That's what you're supposed to see. This is the results for the methyl red test. So to do the methyl red test, you need the methyl red indicator, which is in a different container here. I'll put these guys over here. And you're supposed to put 5 or 6 drops of methyl red indicator into the original culture tube, the original MRVT tube, which isn't a screw top tube. So that picture is somewhat misleading. It's the tube that has the snap-on cap. You're just gonna put 5 or 6 drops in there. Now, do you shake it? No, because those caps leak. You just put it in there and read it. It'll turn red or it will turn yellow. These tubes have been mixed, but notice those tubes have screw caps on them. Don't do that, don't shake them. So you put the methyl red indicator in, you read it at the top of the liquid. If it turns red, it's positive for a large quantity of acid. If it stays yellow, it's negative for acid. The reason we're testing for the production of a lot of acid is because this medium, the MRVP medium, has buffers in it. They have to produce enough acid to overcome the buffers in order to react with the methyl red. This indicator looks red in the bottle. If there's acid present, it will stay red. If there's an alkaline or neutral reaction, it will turn yellow. Even though it looks like it's going to be red here. Okay. So again, don't shake the methyl red tubes. This is kind of showing you the color that you can expect with the methyl red indicator. Red is acidic, neutral or alkaline is yellow. What's irritating about the methyl red test is when you get tubes that are orange, and it's like, how do you read that? They're producing some acid, but maybe not enough to be positive. This is showing the color change for the VP test. So you do the VP test first. You have to take out part of the culture to do it. If you accidentally put your Barrett's reagent, alpha-naphthol and alcohol, and 40% KOH into the original culture tube, do you think you can read a methyl red test then? Why not? Because you just put in 40% potassium hydroxide. You made that exceedingly alkaline. You're not going to be able to read a methyl red test. So make sure you take part of it out to do your VP test first. Okay, so that's MRVP. I'll put the information on the board telling you how to do this, although it's in the lab manual. Okay, so the next test we can look at is the esculin hydrolysis. This was not a test that everyone ran. People that did an esculin hydrolysis test had gram-positive coxy on blood agar. If you have gram-positive coxy on blood agar, you were supposed to inoculate esculin, and it's not bile esculin, it's regular esculin. There are no demos up there. Does anybody have an esculin? Oh, here's one. Okay, so this is our esculin medium that has a positive reaction. So these organisms can hydrolyze esculin. We know this because the medium is now black. The esculin medium starts out a kind of a gray tan. Does anybody have a negative one?
I do.
Oh, there's one. And you do too. Okay, so here's an esculin reaction that is negative, and here's an esculin reaction that's positive. You just look at them. Now I know you have the wrong culture. Oh no, where did I get this? Okay. So this is showing you the color change, and this is talking about the worksheet. So this is on page 194, the data sheet. The medium is esculin agar. Sometimes there's bile salts in it, but when we use this test for semester project organisms, they almost never want bile in there, and so we just stop putting the bile in. There are some tubes of esculin that have bile in them, don't use them. So the initial color is kind of a tan color. If the organisms can hydrolyze esculin, the medium turns black. The next test everybody should have done, most of you should have done, was the amino acid decarboxylation. This is described on page 196. There are two amino acids that bacteria can decarboxylate fairly readily. One is lysine and the other is ornithine. For this laboratory, the only medium that the lab techs prepare is lysine, but the two would work the same way. So we're looking at lysine decarboxylation. An amino acid always has an amino group at one end and a carboxyl group at the other. If the bacteria remove the carboxyl group, so they do a decarboxylation, what's left is a substance called an amine. And amines are alkaline, they're volatile, so they tend to go out of the liquid, and they smell bad. Actually, you can kind of get an idea of what they smell like by looking at their names. So cadaverine and putrescine, the really bad smell that you associated with decomposing organic material, specifically decomposing protein, because the bacteria take apart the protein and then the individual amino acids, if they can decarboxylate lysine or ornithine, they can produce cadaverine or putrescine. So if your organisms are decarboxylating lysine, they're producing cadaverine. It still smells bad. So this involved two tubes. You had one tube that had lysine plus glucose, and you had another tube that just had glucose. The one that only contains glucose is called a control. Again, this is a test that is used to identify organisms that are gram-negative and fermentative. You cannot use this test the way we've set it up if your culture can't ferment. So if you were to find out you had a respiratory organism, don't run this test. It's not going to work. And the reason it's not is because we seal the tube shut with Vaspar. So here's our work study guide again. This is on page 203. It says the amino acid being decarboxylated is lysine. It tells you that the end products that are formed are an amine and CO2. So if lysine is decarboxylated, the carboxyl group is going to come off. That's going to form the CO2, and what's left is the amine, cadaverine. Smells bad, it's volatile, so we have to seal it in solution. That's why we put the Vaspar on. The pH indicator that's in the medium is brome cresyl purple. So you saw this before in the BCP lactose plates. Now, notice the color change. Brome cresyl purple is purple when it's neutral, and it's purple when it's alkaline, and it's yellow when it's acidic. We put a Vaspar seal on both tubes. Actually, I did. After you inoculated, I put the Vaspar on. The Vaspar does two things. It creates an anaerobic environment. So if the organisms can ferment glucose, they will. The other thing it does is keep the volatile amines in solution, because otherwise they bleed. We don't want the cadaverine to get out. But that color business presents a bit of a problem. This is what our medium starts with. This is what it looks like to start. If the organisms can produce cadaverine, it's alkaline. How can you tell the difference between the start color, which is purple, and the alkaline color, which is purple? It's a bit of a problem. Consequently, we have to have two tubes. We have to have a tube that doesn't have lysine in it. That's your control. Okay, so we have a control tube that just has glucose in it. We already know your cultures can ferment glucose because your OF tubes turn yellow. So if they can ferment glucose, they should produce acid under the Vaspar seal, and your control should be yellow. Sometimes after a period of time they'll look white. But if your control is yellow, this tells you yes, they can ferment glucose in that particular medium. You didn't kill them accidentally. So the test result in the other tube is valid. Can I have these up? So here we have a control tube, lysine control, I believe that's yellow. It's kind of a whitish yellow. But that means they fermented the glucose, they produced acid. They also produced some gas under there. This tube then tells us what's in there. Because both of these tubes contain glucose, they can ferment and produce acid from glucose, which means if this one's purple, what's in there? An alkaline amine, cadaverine. Cadaverine is making that tube purple. That means this is a positive lysine decarboxylase result. Okay, so you have to have this combination, your control yellow, which tells you yes, they can ferment glucose, and your lysine tube purple to tell you yes, they have decarboxylated the amino acid lysine and produced cadaverine, an amine that is alkaline. And it couldn't get out because the tubes were sealed shut. Sometimes the purple is kind of faint. Sometimes the yellow is kind of faint too. Yeah, if it's really faint, if the color's really faint, hold it such that you're looking down through the liquid against a white background.
Because there's like purple on top.
Don't worry about that. So the purple on top is because when I put the VASPAR on, sometimes it splashes up on top. That's just the color of the medium to start. Ignore that. Okay, so again, this says, is acid present in the control tube? Yes. How do we know? Because it's yellow. The control tube turned yellow because of the acid. Is lysine being decarboxylated? Yes, because the lysine tube is purple. That purple indicates an amine, alkaline. You have to have two tubes. You have to have a control. You have to have lysine. Okay, the purple colored lysine tube with the yellow colored control indicates lysine is decarboxylating. It's lysine decarboxylation positive. There's gas under the VASPAR. It could be just CO2, don't worry about it. Now, if your tubes look like this, is acid present in the control? Yes, because it went from being purple to being yellow. Is lysine being decarboxylated? No, because there is no amine in the lysine tube. It's yellow. The only thing that happened in there is glucose got fermented. Both tubes have glucose. Glucose got fermented, we got acid. There's no amine in there. There's no alkaline end product. Okay, so that's a negative lysine decarboxylation. Yellow control, yellow lysine means they cannot decarboxylate lysine. Even though there's gas under there, that doesn't tell you anything other than they can produce CO2, and they can do that just by fermenting glucose. Now, if you get this kind of a result, a purple control and a purple lysine, is this a valid test? And the answer is no, because you don't know what's going on there. Was it purple because you forgot to inoculate it? Was it purple because you thought you picked up the organism, but your loop was too hot and you killed them when you put them in the tube? Is it purple because that culture can't ferment anything? The respiratory culture? If you run the lysine test on a respiratory culture, you will get that result. They can't ferment. So that's an invalid. If you get one of those and you thought you were using your fermentative culture, you weren't. Or maybe you were and you killed them. Anyway, you need to do it over again. Okay, next test we need to look at is the citrate utilization test. This is just a tube that's a blue tube with a slant in it. Again, we're not looking to see can these organisms run the Krebs cycle, which would break down citrate. We're looking to see can they move citrate across their cell membrane. We're testing for the presence of an enzyme called citrate permease. And the function of the enzyme is to bring citrate into the cell. If they can take citrate in, they're going to do some other reactions relative to the materials in the medium, and they're going to end up producing ammonia as a waste gas. The information about this, the worksheet study guide is on page 203. The enzyme we're testing for is citrate permease. The product that's going to be formed if they can pull citrate into the cell is ammonia. And the pH indicator is bromothymol blue. So it's the same one that was in the T7 medium, the same one that's in the OF medium. The only difference is that we're not going to see acid production in the citrate tube. We're looking for the production of ammonia, and that's very alkaline. So we're looking at the green to blue color change. Here you see two tubes. The medium starts out green. If the organisms can remove citrate from the environment, they can take it through their membrane, your medium will turn blue. And it's a nice deep blue. Yeah, here's one. Citrate permease positive.
Yeah, green.
It's blue. If they can't do this, medium's green. Unfortunately, the green is kind of a dark green, not a yellow green. So if it's green the way it was to start, it's negative for citrate utilization. The next one we're going to look at is urea hydrolysis. The urea hydrolysis. Urea is formed, it's a breakdown product after organisms take apart proteins. They can then make urea from the various amino acids. But if they can hydrolyze urea, they do that using an enzyme called urease. If they use urease, then they're going to break down urea and they're going to form ammonia, which again is alkaline. In this case, you were supposed to just streak the slant surface of the urea agar. If they form ammonia, they should cause a color change to hot pink. So this medium starts out a little on the acid side of neutral because the pH indicator is phenol red. And it's kind of peachy to start with. Phenol red is more red to start if it's really neutral. So they start a little on the acid side of neutral. But if they can produce ammonia, that color change is going to change to hot pink, kind of a fuchsia color. Here's one. Hot pink. This is urea positive or urease positive. They produce the enzyme urease. On the tubes, if you have physiological unknown 16 or 25, those urea tubes are predominantly peach. There's a little bit of alkaline surface on them. That is because they were sealed in a plastic bag, and both of the cultures produce citrate permease. So there was enough ammonia produced in the bag to cause the top of the agar to change color. But unless it looks like this, hot pink throughout, it is not a positive urease reaction. Also, if it's yellow, it's not a positive urease reaction. So the tubes start neutral. They can stay neutral or they can turn yellow. Both of those are a negative urea hydrolysis. If they do this, that's positive urea hydrolysis. It has to be hot pink. So here you see the color possible. Again, the start color is the peach. That's the color in the middle. They can either stay peach or turn yellow. Either way, that's negative for urea hydrolysis. The only way they're positive is if you get the color that's shown on the right. So they have to produce ammonia, and ammonia will cause that pH indicator to turn hot pink. Some of you did the coagulase test. By the way, for recording these, it's often useful to put them on a white background and take pictures of them. So it used to be students had to carefully draw them out. Now you guys, everybody's got these cell phones, you can just snap pictures of them. Anyway, coagulase test was in the little bitty tube, so they were in that white rack that was turned upside down over here on the counter. They're orange. They have a C on the top for coagulase. This was used for cultures that were gram-positive, and they were originally on mannitol salt agar. So not everybody had this test, but you need to know about it anyway. So, the coagulase, notice the ASE ending? That's an enzyme. What do you suppose a coagulase enzyme would do? It would coagulate, right? In this case, it's going to act on blood plasma, and it's going to cause that blood plasma to become solid. Don't shake those tubes, by the way. The coagulase tubes, to read them, what you're supposed to do is just turn them upside down. Okay, so the enzyme being tested for is coagulase. The medium that we're using is rabbit plasma. So we put the culture in there. If the liquid becomes solid, it's coagulase positive. If the liquid stays liquid, it's coagulase negative. To read them, again, don't shake them, you just turn them upside down. Turn it over. if your liquid is now staying in the top of the tube, just like liquid won't, then it's solid, and that's coagulase positive. If it falls into the liquid, it's still liquid, and that's coagulase negative. But don't shake it, because you can break that coagulation, and then the whole thing is, well, basically you have to start over and do it again. This has to do with new media. So I'll tell you about this new media, and then I'll start putting stuff on the board. Okay, so from last time, you had citrate, urea, lysine control. Everybody had those, and MRBP. So you have to do the MRBP test. You have to do some things to it to read it. You can't just look at it. Citrate, urea, lysine control, you just look at it. Some people had asculin, you just look at that. Some people had coagulase. Turn it upside down and look at it. The new media that we're going to inoculate tonight, primarily carbohydrate beets. There are 9 of these. So we have 9 different carbohydrates, so we want to know, can the culture ferment that? We know all of these cultures can ferment glucose, because we saw it in the OF media, we saw it in the lysine control. They can ferment glucose, but we don't know about the other carbohydrates. So we're going to inoculate a whole series of 9 carbohydrates. You do this using your wire loop, pick up a visible amount of your cellular material and put it to the bottom of each of the 9 carbohydrates. Probably the most important thing to remember about our carbohydrate deep media, they all look the same. The baskets are going to be up here on the front. Don't take the tubes out without having a way to label them as you pick them up. Because if you pick up a handful of tubes and you get back to your desk and they're not labeled, they're worthless. They all look the same. So you have to label them as you pick them up. And then you inoculate by just stabbing. Another medium type we're going to inoculate is called the SIM medium. SIM stands for sulfur indole motility. And if you really want to drive me nuts, refer to this as the SIM test. There is no such thing. This is sulfur indole motility medium. It's 3 tests in 1. So it's a relatively large, deep. You're going to stab your culture to the bottom and try to go out on the same line. So you want to make sure you use a straight wire, put it to the bottom and out. These are really low, but that's what we have. So this is SIM. It's going to be testing for 3 things. Do they produce hydrogen sulfide? 2, do they produce indole? And 3, are they capable of motility? This is a 3-part test, 3 different tests, 1 medium. Sulfur indole motility medium, or SIM. And the 3rd thing you're going to inoculate is triple sugar iron auger. This is abbreviated TSI. And although students often refer to the SIM test, they very rarely refer to the PSI test. Neither one of them are to be read as words. They're acronyms. SIM, 3-part test. TSI, multi-part test. TSI stands for triple sugar iron. So there's 3 sugars in here. The sugars are glucose, sucrose, and lactose. The iron in the medium is testing for hydrogen sulfide production. See this little white spot? That's an indication of where you should stab. So to inoculate the triple sugar iron medium, again, use a loop. You're going to pick up a visible amount of your bacteria. You're going to streak from the bottom of the slant to the top, fishtail, back and forth, and then put your loop about there and stab it to the bottom. If you try to stab to the bottom from up here someplace, you're going to put the handle of your loop into the medium, and you don't want to do that because it's not sterile. Okay, so you're going to inoculate tonight. 9 carb deeps, TSI, SIM. Some of you are inoculating everything. So, things to do. You're going to observe and record the data from last lab. Take pictures, that's a good way to record it. Then you're going to get the media for the new stuff, and you're going to label it very carefully. And then you're going to inoculate your new media, and you're going to put it in the baskets again. We'll have to get new baskets. They're going to be labeled so that you're, and remember how you had to put the little tape labels on top? Please do that again, because it makes it a lot easier to find them. And then that last part, put PCR product into a gel. We are so behind. We don't have any PCR product. However, we do have a couple of cultures that can be processed, that is, DNA extraction. So Leah, we can do yours tonight. That, there should be a plate in there that was inoculated Sunday, maybe? Yeah, over the weekend. Anyway, if you have a semester project culture that you've been working with, please streak a new plate tonight so we can extract DNA on Thursday. We need to get these projects going. If you've been kind of hemming and hawing about a semester project and you just don't know what to do something. Use an air plate, use a soil plate, I don't care, but do something. Don't just let this wait, because you're gonna run out of time. Nobody's running gels, we don't have any PCR product.
Change your background.
Dang. I got accepted in state. Diversity.
I don't even wanna look.
Congratulations.
How'd you figure that out?
They just emailed me.
Like your Sierra?
Yeah, no, that's whatever email you use for
I haven't gotten any emails. I didn't get this last day.
You use that email? Is that over your account? Are you?
Okay, so as I'm putting information on the board, the first test you need to run where you actually have to do something is the Vogt's cross-scour test. The screw top tubes are in this rack and the caps are in a different, they're in a different basket. Some caps fit some tubes. You have to kind of mess about with these to find a match. Anyway, tubes and tops. First thing you do is transfer 1,000 microliters into the screw top, and you add 18 drops.
I'm not gonna just stand there and river. Where did you get that in?
Over the weekend? Why is this one so dirty?
Be careful with ferret reagents. It doesn't matter too much because you're testing for a Sedawan and it should just be in the case.
Rosie, do you need one too?
Yes, please.
Ariel, do you want to leave?
I'm really impressed.
That's what sticks with you.
I know, but like 40
It was
billion
dirty.
people have probably touched it today.
What tube? Which one are we supposed to use for this?
Like, stress here. How was it sound? How was it? Sunday, right?
Well,
No, yeah.
I'm reading an article so today I'm Reuters.
Yeah, you can't
What are they gonna
inoculate, check, you have to inoculate and let it grow.
do? And you know what? You know what I did?
There's nothing that was, there was a racket.
Or they're gonna walk that room. Like that's the thing. Like, you know, it's absolutely not. It's some that are like.
Don't you check your tube to make sure that you don't have the cell tube in your rack by mistake? Her MRBT tube is missing.
Farm in Fall River.
Those little people
A farm in Fall River. I was saying that. But now I'm thinking maybe like.
What'd you do, Rosie? Hers won't come off.
To get the tip
They're
off
clicking all the way
our
down, it's
both
not coming
thumbs.
off.
I don't know what we're doing. What are we doing? Do we all need like? Rosie, do we all need one?
Yeah, if you get the,
Yeah, because you have to do.
if you have it.
Do I have what?
This one, the MRBP.
And I put that in here. What are you waiting for? Just to clear up?
Yeah, to get one that
Just type that thing in here. Anything else I can be doing?
I feel like I shouldn't even do it. If my whole thing's off.
Sure, ask her.
I'd call it negative. What number is it?
I'm wondering if
Did you find out?
It's not either in the box or in a drawer.
Yeah, as long as you don't lose your.
I said, now you need to start writing my own project, but I don't think. I think I might. Probably. Oh, yeah, I got it. Ooh, that's really good.
Okay, that's not like that. Let it come up. Oh, sorry. I thought you were.
The one first page and one first. Everything on the ground.
Yeah, but I only do the fermented.
put a thing on the top. About up here, your MRVP, your lysine control, citrate, urea, escalin, etc. You can't keep them because we don't have enough racks to hold them. So what you do with them is remove the tape labels and they go in the plastic racks down here in the discard area. You must part with them. Please do not leave tape on them. The people that have to wash these tubes do not want to remove hundreds of labels. If you leave your tape labels on, you end up on the wall of shame. You don't want to go there.
It's a lot of people.
It'd be a shame. So it doesn't matter.
Where's the one I just made? Where's the one I just made?
Austin, take your labels off.
Can I use your
Oh
yeah.
One. I haven't put my drops in this year.
Right.
Looks like. When you put your drops in.
What's that?
My thingy.
You haven't put the drops
Your
in.
MRT or MRVP?
Where's the dropper? Is it on the wooden board? No, it's what DJ's doing right now. Okay. Move over, DJ. Come through. Get in the way again.
Oh, that one's right. Mine is not.
Mine's still on there.
You're an asshole. Gotta have the timing.
Oh, crapola. I took the MRVP from. I gotta do that again. I have 2 MRVPs.
Why do you have two?
Because I had to do it for both. I'm just gonna do it again.
Alright. I guess you can dump all mine now. So my MR is
Then
acid.
I did 2 MRVPs and I did the
My VP is yellow. Can we stop this?
It is no. Just in case. Just in case we have to press play again. Not to have more than one video.
It's already going right now. It's like
I know, that's why I said shut.